So, a very warm welcome to everyone. Thank you so much for joining in. A very good evening to all of you. My name is Diksha Bhartiya. I'm your host for the day, and I am here on the behalf of our company Genomics. We are a new venture. We are working towards providing bioinformatics services and developing solutions for life science and healthcare. We have been incubated at NSR Cell at IIM Bangalore. So as I've told you, genomics is aimed at providing bioinformatics services and solutions for life science researchers and healthcare industry. So the different key competencies that we do have are providing bioinformatics support, which include mostly the next generation sequence data analysis support, various structural bioinformatics support, and the different other training programs that we regularly conduct, including workshops and webinar and short and long-term hands-on training sessions, including dissertation and courses, etc. If you are looking for any sort of trainings or any sort of data analysis services or any sorts of solutions, uh, feel free to write to us. If you have any specific or customized requirement as well, we'll be very happy to assist you. We'll be very happy to design a course according to your needs. Or if you have a customized or meta analysis that you're looking for, don't feel shy. Please write to us. You can call us directly or connect to us with connect with us on any social media platforms. We are quite active on LinkedIn and Facebook and all other Instagram, all social media platforms. And the different courses that we do conduct are the ones mostly covering genomics based subjects. NGS analysis is the one of the major course that we regularly conduct. The other courses include the basic bioinformatics, structural bioinformatics, and we also have another sub course for the vet lab bench biologist, which is called the exposure to the bench, where we teach about the tools of the lab. We a few weeks back we conducted a course on genome editing as well, and where participants were taught how to about the basics of the genome editing and how to design CRISPR-Cas RNA. So basically, and due to the pandemic, all of these courses are virtual, and we do provide the certificates and the recordings. So let's now move on to introducing our speaker for the day, Nitish Kea. And he has done his bachelor's in life sciences and he's done his master's in biomolecular and biomedical sciences. He has his initial research experience to publication of COVID-19, where he's done a bioinformatics analysis and a microbial polymer production and its application from a novel organism. He has also studied on COVID-19. He has also done a COVID-19 publication on variants of the genomic material for identification and better treatment of COVID-19. So basically, today he's going to use his expertise and his knowledge and his experience of performing research from the very basics, from his own idea. So if you have an idea, how do you convert into a proper research? So that is he going to talk about. So he also got this particular idea at Glasgow Caledonian University through Santander Enabling Entrepreneurship Fund, which enabled him to work on bioplastics, and which have where he has continued to work to obtain a patent and has now registered a venture called Microbiome Biotechnology, which is an LLP, and it's also incubated at IIM Bangalore, NSR Cell at IIM Bangalore. So a very warm welcome to you, Nitish. And his co-founder at Microbiome Technologies, Vismita, is also among us. And so we are very happy to have you here on this platform to talk about the various fundamentals of research, where you'll be talking to these students and the participants about the meaning of research. How do you define the research? What should be the different objectives, and how can they define a research problem? So if then they have an idea, how to proceed from that idea and make a particular research proposal, they have a complete process to make a research proposal. And I'll be very fortunate to have you on this platform. And we'll be very happy if you can also talk about your experience, that how you got that idea of the bioplastics, and which gradually enabled you to make this proper venture called Microbiome. So, and thank you so much for accepting our invitation as well. I have stopped sharing my screen, and I'll make you the uh, uh, enable your screen sharing now. I've made you the presenter, and And the participants, if you want any questions, you can ask them anytime. You can write them down in the Q&A box, which is there on your left hand side. And if you want yourself to get unmuted, you are feel free to raise your virtual hands so that you can we can unmute you and you can ask your questions right away. If there are several questions, we can take them at the end of the talk as well. So, I also noticed that some of the participants were joining from two devices. I would request you to please join in from single device. And yeah, now I'm done. So over to you, Nitish.
Thank you, Diksha, for the introduction and for the opportunity of helping me to, provide the information, the basic research that I have gone through for the students or interested candidates who are willing to do research at this point of time. So I will share my screen. Please do let me know if you're able to see my screen.
And Nitish, I would also like to bring to your notice that most of the participants that have joined in today or who are registered are in their masters or bachelors. So most of them are doing their BTechs in biotechnologies. Some of them are even in their BScs or BScs. So all of them are students, basically young students who are looking forward to understand how to how to do a research, how to get into research. So basically, I understand from the perspective that since they are in a very initial stages of their career, so they're trying to understand that if they are planning to go into a research career, so if you can throw some light on that as well, that will be really helpful. Nitish. Nitish, Vismita, can you hear him? I am not able to hear him. His screen just.
Yes, you are audible, Diksha.
But Nitish is not audible, right?
Yes.
His screen.
He's reconnecting back.
Yeah, I think he's reconnecting. So just give us a moment, participants. I think there's some network issue with Nitish's end. Yeah, we can see you now, Nitish.
Yeah, I'm sorry. I don't know what happened. It just broke out.
Yeah. So yeah. Now you can share your screen. I have to make you present. Yeah, you are already a presenter.
Yeah. So you were saying, sorry. So you were saying that most of the students are bachelors and masters, right?
In fact, 80% are BTechs.
Okay.
Yeah. So you have, so you should understand that the crowd is, at a very nascent stage of their career. So
yeah,
in the biotechnology.
Yeah, I got it.
Yeah, great.
Just a second. Yeah, can you see my screen now?
Yes. Just make it full screen.
Yeah. Is it full screen?
Yeah.
Okay. So, hello everyone. I am Nitish, as Diksha gave an introduction. Today I'll be going through very simple thing, the fundamentals. As we all know, that research is an scientific investigation methodology that we go about. But in your process of being at masters or at bachelors, I know you would be in a state where many people are giving you instructions like this is how you're supposed to go, this is how you're supposed to go, and you will be like in a space where you're confused, like the black arrows that depicted on my slide. So what happens is, what is the perfect methodology, or rather than say an ideal one for you, catered in such a way that it takes you in a process and orderly manner to give you out a complete structure how you're supposed to go about it. So the first thing is that you have to identify an idea or a problem. So when you say idea, the initial thing, idea is something kind of become a fashion word into an innovative part, but it is not as easy. I do accept that everybody is not Einstein or Thomas Alva Edison or somebody else like them to get ideas out of nowhere. Like you just pop your fingers and everything comes to you. No, it doesn't happen that way. So for you to understand, to get an idea as such, you need to inculcate small habits, say like going through your recent technology. And now you might ask me how. You can do that through by getting in touch with Hindu newspaper. On every Sunday they do publish a section of science and technology, where you will be able to approach all the new recent happening innovations or studies and research that is happening in the country and outside the country as well. So that would be a very much short space for you to connect immediately at whatever stage you are. And the other general things that you might know as your teachers and peers or lecturers, everyone would be saying you is that libraries would have certain amount of your journals, and you also have databases. So generally, what every student does is, if they need an article on a particular subject, they directly pop into Google. Google is a very large database, or rather, to tell it as technically, it's a search engine which will pop out information from every bit of the source it can get you. So the major thing as students you need to focus is the databases that is present, such as eJournal, Science Direct, or Web Sciences. All of these databases that I have provided in this slide is, it provides you direct, specific, and more subject-oriented information for you. So now you might ask me the next question. Okay, I might have a habit of this, or I will inculcate a habit of this. So how do I go about this? So let's take an example of John Gregor Mendel. I think everybody knows him, right? So he's one of the famous person known as father of genetics. How do you think he became the father of genetics? So everybody, as you know, in textbooks and in your classes, you have heard, you have read about him. But the basic thing every teacher fails to tell you is he was first observational, right? He was observational. He was not any specific science graduate or any other graduate. He was just a monk in a church who was going, just like having everyday walk and talk with his peers and all his chores. But one thing that struck him was his observation. The pea plant that were grown in a small garden kind of a setup near the church attracted him on a daily basis, because he started noticing certain small features which made him to understand, okay, there might be something like, say, sorry, there is a height difference. Why is height difference? Because every plant is coming out the same seed. Or say, consider the same plant is coming out of one particular plant that is giving out the seeds. Then how is it getting the change in the height or the number of tendrils that it is producing or the number of leaves? So he had this question, but he did not know how to go about it. So how do you think he went about it? The simple way that he thought was he should first approach a botanist. So that he did. And the first thing he asked to them was more about its reproductive cycle. How does the plant reproduce and how does it go about its reproduction? So then, with the information from the botanist, he understood that flower is the major reproductive part, and that could help him study the plant even more better. So from there, after understanding the functionality of pollination or the reproductive cycle of a particular plant species like angiosperms, he understood that the plant is self-pollinating. So what do I do now? Then he started to prepare an experimentation, the observational experimentation method, where he created two sets. One would self-pollinate, the other one would be pollinated by him as an unnatural condition, because in natural conditions, it would just pollinate by automatic process. So there, if you observe now, what he did was he first had a background information analysis. Then he worked on observation part of the plant to gain certain aspects. Okay, now the observations that I have done from the previous days that I was walking every day, like maybe the size of the leaf, height of the plant, color of the flower, and all of that. So when you go back and just connect to this point that you are reading daily aspects, right, from the newspaper or the journal or the whatever source you're trying to approach from, what happens is it provides you a brief kind of not exactly everything, but brief connection with the stage of research and development that is happening day to day. So when you are asked to make an idea, you have a kind of a scanner, like how you do for your exams. If you don't have time to study, you go through the scanners, which gives you an overview, right? So similarly, the next thing you would be doing in these articles to collate your idea is finding out specific keywords and streaming it down to what subject it is performing on. The research has been performed. Have they provided any important techniques for which they have approached in the research? Why did they do and what was its importance? Like, you should know why was it happening, right? Sometimes major of us just go through the articles just to gain information, but we never question why did they do it, or what was the purpose and when did it happen, or where exactly it was happening, and then they took it into a research manner. So this, while reading articles, we will need to patternize it and understand it in a very simple form in the questions such as why, what, when, and where. We will learn that with an example. And by learning this, when somebody asks you to generate an idea or a problem statement that you need to work on as your research, it will help you to make certain combinations and understandings that will provide you how to generate an idea. So let's really see whatever I'm saying, does it really work, or am I just blabbering? So this is an article from 2013 and 14, extracted from Hindu newspaper. So if I'll just give you 5 minutes, please go through the article how much ever you can in the 5 minutes. Try and put the key things that I've mentioned in this slide, like keyword, subject, capturing materials. Try and interpret what you can think as why they were doing it, and if you can segregate it into. Let's see whether it falls into the picture as I'm saying it, as I'm saying. So your time starts now.
Shiva, is the presentation not visible to you? Because everybody can see the presentation. Uh, there is a query that presentation is not visible.
Is it?
I can see it. If this is the problem with other participants, please write it. I don't think there is any other participant that has mentioned that.
Yeah.
Okay.
Hope you guys can see the article clearly.
So I request everyone to make this webinar a bit interactive, so that you can uh, get the most out of it. So if you want to speak anything, just raise your virtual hands and we'll unmute you.
If anybody has finished, you can raise your hands and we can work upon it. And please don't be shy, because when you interact, you'll get to know a lot of things other than what I'm just trying to present, right? So please try and communicate. It's okay. Everybody make mistakes. Even I made mistake, but eventually I learned from my mistakes. So please be bold enough to speak up. We just have 40 seconds more. Yeah. So guys, anybody would like to say what's happening? Hello.
I don't see any questions or any.
Yeah. Yeah, even I'm trying. Yeah, there is one person raising hand. Amrita Varshini.
Uh, Amrita. Yes, great. So, Amrita, I am allowing you to talk. Is there anybody who would also like to talk? We can unmute you. Yes, Amrita, you can go ahead.
Am I audible?
Yes.
Yeah. Yeah.
Okay. I was able to figure out a few keywords from the article as such. The nanocapsule vaccine, that would be an important keyword. And HIV, the infection for which they tested this out, would be another keyword. And I also thought the model organisms, right? They used mice. So I thought that could also be another important keyword.
Yes. Yes, that's great. So did you feel any difficulty in finding out? See, because the keywords that you found out, match very much specific to, as I said, but did you try why was it happening? Why this kind of specific research had to take place?
I realized that if we have to, try to understand and process what the article, is trying to mean, it would be much easier to look at the keywords and, as you said, the subject, and interpret it based on the WH questions and all of that.
Yeah, yeah. Okay, I think you're on a very good track, Amrita. So I'll just go in the next slide and let you know what exactly you missed out. Anybody else, guys? Because whatever Amrita was trying to find out was, kind of simple. She's getting into the thing. If anybody has at least done the same thing, you can just communicate to me. Okay, nobody. I think a lot of people are shy to communicate.
Tushar, I've unmuted you. You have raised your hand.
Yeah.
Tushar, you can go on.
Hello.
Yes, Tushar, you're audible.
Okay. Sir, I noticed that the word oval albumin antigen.
Yeah. Great.
Yeah, I think antigen is a very important role in our body.
Yeah.
The vaccination for
Yeah.
antigen and antibody forms.
Okay, Tushar, I think your keywords that you gave are also very much effective. So now let me get back to the presentation and tell you what are the exact keywords and why I have picked it up in such a way. So in the example, the first thing that you need to observe is the heading, right? The vaccination itself in the keyword provides you a huge amount of techniques or methodologies that they will include, right? So considering vaccination as one keyword, you can break it down like vaccination, meaning to say you need to have one specific particular target and one specific particular host to which you're treating out. So there comes your first microbiological steps that you choose up to and how you can go about it. The next thing in the heading itself, you can observe two other keywords. One is inhaler and the second one is jab. So you generally know that any vaccination from your very soon after your birth, they provide you a jab of any kind of vaccine, right? But they have converted this vaccine delivery system into an inhaler. So that is providing you another method that they're trying to process it and their application as well. So the other keywords, as you guys were mentioning, yes, it is immune inducing, the albumin part. Apart from that, there are few more things that you missed out. See, they have mentioned moisture tissue, the mucosa. So here you need to be you need to be very much proactive when you're reading such articles to segregate all of this. I do know that you might struggle a bit because nowadays the courses are more like going into applied aspect where you will not have the minimal background of say the physiology part. If you have, it's really great because that will help you to break down for your research idea that you are developing. So here, the moisture tissue, the mucosa, helps you to understand that the mucosal layer do have lymph nodes, which is very much major part of the immune system that helps to process and progress in your curing of any pathogen infection. It might be a virus, it might be a bacteria, or it might be any fungi, right? And they also have mentioned where all they could see the immune activity, right? They could see the immune activity in the airway. The airway is nothing, your trachea, your alimentary canal, and the reproductive tract as well. This is very important for HIV. As Amrita was mentioning, yes, HIV is one of the keywords that you very well identified. So if you have noticed, the mucosal treatment through nasal has triggered the lymph node network and triggered in the specific area where they wanted to treat HIV, right? The reproductive tract. So the connection of the lymph nodes and their work mechanism, all of this, every time in every article is provided in bits and pieces. It is us who have to break it down and connect it. Now coming part to why they did it in this manner, because they proactively have connected the dots of how immune system is working. It's not only through the general humoral response, it is also having certain segmented areas to protect the body from the oral cavities, right? Like your nasal opening, buccal cavity, and excreta, as well as the reproductive tract. So all of this majorly contain what? The mucosal systems, which are connected through the lymph node system. That is how they were capable of generating an immune response even at reproductive tract but when the vaccine was given through the nasal, right? So now you can say, why was it done? Because such kind of connections were happening in the immune system. And also the other part is that they have to first do, when you say what they did, they first did the initial vaccination studies like general vaccine studies, breaking the pathogen, that is HIV, and finding the antigen, as the Tushar was trying to say. So this is how you will initially start collecting all of your information. So when you go through multiple articles as such, you can generate idea, because this inhaler part is new, right? It is application. So when you read multiple articles, maybe tomorrow you will have to generate an idea in microbiology. Somebody gives you, say your professor or your guide is giving you an organism, asking you to find out, see, the organism is dying in particular condition. Can you say how? So then by reading these articles, you will get a particular set of mind that will help you to put out certain methodologies easily. You immediately don't have to pop out your systems and then search, okay, what is the methods? What are the things that is happening? Why like this only? You don't have to ask it then. But this kind of brief information will help you to generate idea. But in case of problem statements, you don't have to worry. See, problem statements will majorly be given by your professors or your guides who literally want you to work only on that. They don't want you to generate any new ideas, right? So it's easy to get problem statements, but it's difficult to generate your own idea, which you can do through such article readings and connecting on a daily basis. So next thing, okay, now you have generated your idea. What is the next step that is important? The next step that is important is your aim and objectives. I know, it's a very simple thing that when you say aim, everybody would give a huge 2-line sentence and talk about all of it. But aim is just generally describing your kind of, end. Say, for example, let's consider you have to drink milk. Today, I am going to drink milk is your aim. It's not your objective. It's just your aim. How do you drink milk is your objective, right? So let's consider it in a more technical way. So let's consider a titration. Titration is a very simple thing that everyone would have done. So the titration part, the aim mentions that the titration is against an acid and base, and an end point would have a certain color indication. But how would you achieve it is your procedure. That is the objective, right? So that you will be converting into your methods and methodologies when you are performing your wet research. That is how you will be approaching it. So let's consider the same example again and see what is the aim of the example that I provided you, and how did they make their objectives to reach their aim. So the aim is transformation of vaccination delivery mode from injection to inhaler, or a jab to inhaler. That is what the title of the article is also telling, which will be easier for you to understand in your own words when you compare, the analogy of your titration part. It will be easier to break down things and patternize your article for your understanding as interpretation as well. The next thing would be the objectives. See, you can see that it is saying that immunogenicity inducing, right? That keyword will let you the objective to be created as identification of potent antigen and target. That will become the target. So similarly, by going through such keywords, you will get to know what was the objectives that they took upon. Yeah, so now you have generated your set of aim, what you want to do, and you also have a particular objective, how you would like to approach. But don't fix to that objective 100%, because as you know, research doesn't happen as expected, right? If it is happening as expected, then we call it as biased. Biased research acceptance is very much less in any journals or any scientific communities. So be open to modify a little bit when you're going through the literature survey. So now literature survey, how do we do literature survey? Literature survey is not difficult actually to say, but people do complicate it. You know why? Because open Google immediately, type down the whole heading, whatever your guide has suggested. It doesn't work that way. If you type something like that, you know, Google is called a search engine, and it search all the words, whatever you have given in its search bar. That is not the way you search. So you, as I was saying, that you need to make importance of the keywords that is provided in the article. It helps you in your literature survey as well. So now those keywords, while entering into, if you just generally enter it into any databases or any search engines, databases might still give you relevant articles, but Google as a search engine will not give you relevant articles. It will give you a whole bunch of garbage, right? It will not direct you anywhere. Then how do you go about? So what you have to do is you need to use something called as Boolean functions, which is very much useful for you to segregate and save your time around 90%. So these Boolean functions are and, or, and not. So the function and helps you to include two keywords. The function or helps you to either select the first keyword or the second keyword. The not function helps you to select one. Say, for example, I have selected vaccination, not COVID-19, because COVID-19 now in Google has become very much popular. So when you give not COVID-19, whatever else is related to vaccination apart from COVID-19 will pop up in Google. Right? So that will help you to use and, you know, narrow down your search. Now, once you do all of this, how do you select your articles? That is more very much important. So a simple trick for it is, before reading the whole article, please don't do that at all. It's completely waste of your time. So simple technique is that when you're going through Google or through any other databases, beneath the heading, there is 2 to 3 lines of description called, this is called as descriptive analysis. So in this descriptive analysis, you first observe the keywords, whether they're present or not. If they're present, then go into it, then read only the abstract with the keywords and does it make any relevance. If the abstract is making relevance to you, then save down the article, read the whole article later. Even while saving, please save it with the article's title. Don't, or just give it a number, how you can give it, because it will help you in segregating articles later. So let's try with the same COVID-19 literature survey. You can try it at your own time and space, because if we try now for individual, it wouldn't happen very well. Yeah, so when you're creating your idea and aim and objective, the other thing that you need to consider is hypothesis, right? The aim, once you have created through literature survey, where I said please have some space while you are doing literature survey, that is because of this hypothesis. So the hypothesis is the possibility, like how you write in statistical experimentation in a particular population, whatever you're considering as characters. Similarly, in research, a hypothesis is very much important, because if you have a hypothesis, it can either be a positive or a negative aim that you are trying to work upon, right? So considering the same vaccination part and having all of the background information about the mucosal layer, if it turned out not to be happening in the reproductive tract, then what would you do? So to understand all of that, you need to write down your hypothesis, and the hypothesis should be very much specific, like clearly writing down your aim is what hypothesis says. But in aim, you will have to add 2 lines, which will convert it into a hypothesis, saying that specific and whatever you have isolated from your literature survey. The first thing that you would say, yes, this is a possibility, and this is the methods and methodology that I've chosen to happen, but it might not turn up this way. or it could happen the same way as I'm expecting. So this kind of statements will help you. This is just for you. Don't put it out to any proposals or anything, right? For them, it's not the appropriate way to deliver. So we'll see that in the end how we will do that. But for now, you need to understand that you, for your individual analysis itself, you need to consider yes, no, or an alternative method that could help you to understand your research. So this will happen simultaneously when you're doing your experimentation and when you read your data, right? There again, your hypothesis plays a role. So we'll see it how it goes. Once now you have done with all the literature survey, hypothesis and everything, you should understand how your methods and methodologies will suit well while considering the samples. You see, actually for me, when I was doing the bioplastic, I never had this kind of sample designing at all. So it was like whatever I do was right. But once I had been working in a private lab as Zymes Biosciences, there a PhD person came and asked me, what exactly are you doing? So I was explaining him. So this is how the steps I was taking, like the sample I have taken it for cell culture study, meaning to say the growth cycle, and then analyzing its polymer production. Then he was saying, is this your sample design? So then I had a question. What do you mean by that? There, I got a learning that you have to pick up samples in such a way when you're doing a study that it suits your methods and methodologies that you've selected. If not, you have to design it in such a way that it falls in it. So let's take an example for it to understand. Say we are doing a phytochemical analysis, or okay, let's make it simpler. We are just studying one of the antibacterial property of one particular antibiotic produced by Penicillium notatum. So how do you do that? Here, you can't pick up a random sample, because a random sample comes into a sector where you have something fixed. But the variance happens only in that fixed set of the sample. It does not happen in outside the set, right? So here comes your more focusing down into how you treat your sample. So when it is more like statistical part that you're understanding, considering only one kind of population and seeing their eye color as in studies for genetics, then it's kind of fixed, but you will only see the variance that is happening. So there you can do random sampling. But here you don't know what you're expecting at all, right? Antibacterial property, yes, that is one expectation, meaning to say it is killing the bacteria. That's a general theory, but it does not provide it as a random sampling or the set of probability to work in this kind of genera or phylum only. You don't know that. So to understand that, there is no particular probability that you can expect. Say Pseudomonas aeruginosa can be inhibited or non-inhibited. Maybe tuberculosis bacteria like tuberculum, I'm sorry, I forgot forgetting the name, but the tuberculosis bacteria can be inhibited or not inhibited. They both belong to different genera, phyla, and all of that. So there you can't make a probable expectations. So that is when you will not be following a particular random sampling, but you will find out the methods like ABST, that is antibiotic sensitivity test. So that is your preliminary test observation to understand, yes, this is inhibiting this bacteria. If it is inhibiting this bacteria, then the next thing what you will be doing is your concentration analysis, right? See, so now by understanding the first sample of the bacteria which to pick, you actually got a line in order. Okay, first I would do ABST, then I would do concentrations, then I would go to understand whether it's happening onto the surface level or is it happening onto the molecular level, is it just blocking the cell wall. So it follows step by step by step. So that is how you need to pick up your sample. Otherwise, it makes a lot of confusion when you don't know whether you're doing a set of well-known information or set of just general information. That is where you will be designing your sample, which will help you to flow the methodologies that you might have selected previously. But once you select your sample and it falls in flow, it gives your research a very good flow without any hindrance. So that is what, considering the probabilities, that is what Gregor Mendel did. The variance, what he was trying to do was happening only in a particular set. Say, for example, if he didn't know the probability, he just tried to pollinate the pollen grains of tea plant with a hibiscus. Do you think there would have been production of seeds, or the vice versa way? No, it wouldn't happen, right? So sample design helps you to go about the methodologies and the restriction of methodologies that you might have when you're performing your research. When you're performing your methods for the idea that you have generated, the first thing that you generally would have seen in your bachelor's, that you must maintain a record for your lab. They actually don't do it on to torture you, but it's a kind of, you know, scientific journal, what they call. For us, it happened to more to be a record, you know, the general term record. But it's not a general record. Even, for example, I can give you one simple instance. When I was working in my bachelor's in the lab, the data that they had generated for the recombinant DNA technology for producing beta-galactase enzyme was completely different each year when they had performed. They never noticed it because they were just giving the previous records to students and just copy and go, leave it away. They just used to sign and you will get the marks. But what I noticed was there were almost 2-digit variants or 1-digit variants in the colony formation, the enzyme production, and the experimentation methods that they had used. So this will help you a lot. So never ever miss your data inputs into your scientific journal or the record, because this will later help you analyze if you have to reassess. The other important part is that will it match to the data what you are, you know, kind of processed in your literature survey? Is your experimentation going smooth, or, you know, kind of when you made the hypothesis it is not possible? Is it happening towards so much? You will get a brief idea when you are writing down all of this data and observing it, right? So the data might also differ when you're considering like experimentation as Mendel is doing. It's just an observational experimentation where you just write down the features. But in experimentation such as antibiotics, it's not only just the observation, but you're also performing certain aspects such as molecular mechanism that might be happening inside. Through your literature survey, you're just connecting it, right? So it's completely different. So data information you're never supposed to miss. Once you have completely finished your methods and methodologies and recording all of the data that you have generated through your experimentation, now the main thing is that you're first supposed to sit down, read the whole data, see whether it is matching to your hypothesis, right? This is where hypothesis again comes into picture, and understand, yes, it is falling into more the way I expected, and it is giving a particular result. So if it is giving result, then it's great, well and great. You don't have to delete or add anything extra. But in case if it is somewhere in between, then you might have to perform or maybe add another methodology. So before writing a report, when you have all of this data, you just need to reassess, because that will help you again to make your research solid and just not on an overview like the water bubble staying on the water, right? It shouldn't happen that way. If required, then you can add upon your statistical analysis. There are many statistical analysis like t-test, f-test, ANOVA, and all of that. But that is significant only in case of probability. That is the set of information is very much expected. The variance or genetic studies you're doing, only in such cases. In case of not expected results, you can actually make certain modeling, like this kind of concentration killed this kind of bacteria. So this has to be tested in a concentration mechanism. So you can make a modeling there. For example, considering a mice for an in vivo studies, or say any other minute, let's consider the most minutest model organism, that is Drosophila melanogaster. For it, how would you treat? Because the same concentration that you're treating would not help there. So you can give a brief discussion kind of part in your research, which will help further analysis of the antibiotic that you have studied in your research. So understanding the same thing, if you put the concept into the same example, the concentration part has played major roles. And if you read the article, you will now get to know how you can process the data and how it will also help you in idea generation, methods and methodologies, and making it whether reassessing or not reassessing, right? The basic parts that you have to do in your process of performing the research. Yeah, this hypothesis testing is more related to the statistical part. If you're not using statistical part where the probability is there and some cases might not have, so you can just skip this. If not, the hypothesis that I mentioned initially is different and this hypothesis is different, right? In this hypothesis, you're trying to prove whether it is significant or non-significant with all the statistical equipments that you have, like the test of t-test, chi-square test, f-test, and ANOVA as such. Yeah, the final and major part before writing a report, you have the data, you have analyzed your data, now you have to put it in the form of verbal part. How do you do that? So in this part, it's very, very simple thing that you have to first generalize the subject. Meaning to say, don't just bring it to the textbook level. Please don't do that. The simple thing that you can do when I say generalization, you have a literature survey which gives brief information about how they came into effect, as what treatment happened, how it was, by particular methods and methodology that they selected, how they understood the molecular mechanism that antibiotic was killing the bacteria. So having such thing, now you can generalize your studies without doing or performing the same X-ray or the electron microscope studies or the powder X-ray diffraction and all of that. You can generalize it by saying that when we treated, we could see the inhibition zone and so and so. So the expected part is that the antibiotic is still playing a role in the molecular level to kill the the enzyme activity and to also break down the cell wall with osmotic pressure to completely inhibit or nullify the effect of the pathogen, something like this. So you're generalizing. And from the data that you have given, the numbers, the quantity and the quality numbers that you have, you can put in that numbers there. That will start increasing your stability as well as your data part, which is coming into the verbal part when you're writing your results section, converting it to the verbal form. Also, please make sure that the literature survey that you have plays a part in your generalization, because that will clear, the gaps and bridges that you might be running inside your head. It makes a proper circling and connective parts, which will get you into the right direction of writing your interpretation in report. And also please be speculative about what you're writing. Don't write it only just because you did it, right? Write it in a format where not only you, say for example, if I ask you, how do you fulfill your hunger? You have basic explanation saying that I am hungry if I eat food. my hunger will be fulfilled. Similarly, your research has to be conveyed in a simple fashion, or your vocabulary could be very good. So that's great. But that should still give the connection to the reader who is trying to read your article or your research, right? This kind of interpretation and putting it down into the verbal form will help you to very clearly depict what you are trying to do. In report writing, it's general, as you know, writing. Yeah, but in this report writing, no, the first thing is that everybody would say write abstract. Please never do that. Writing abstract at your initial stage is the dumbest part that you can do. Just follow about through your literature survey, go about the introduction part. Methods and methodologies you would have first generated, but you can add an addition through your literature survey. Then you do your experimentation, write the results, tabulating part, and all the image and writing the conclusion. After writing all of this, go through the whole part of research that you have done. Now summarize it. That will become a proper abstract when you're writing your thesis in your university. Otherwise, the abstract that you initially try to give is more a hypothesis rather than an abstract, because that hypothesis is you creating inside your head, right? It could be possible because your guide would be delivering in such a way that yes, this is an expected answer. So write down the abstract. Please never do that. Maybe to satisfy your guides, just write it one then, but please do revise your abstract when you're finishing your report writing. Now, yeah, so the synopsis, when I was saying hypothesis, the thing I say that the proposal part, this is what you need to understand. Synopsis is a brief description for any research society that you will be proposing for your research, right? Sometimes it might happen that you are going outside your university. If you're in university, your professors will help you out. So that would be easy and go, and it is easy for them to get you in. But when you go out of your university, how do you put out your thoughts in such a way that it becomes a proper synopsis for your research proposal? The first thing you need to clear out is the first initial part that I was talking about, your idea or the problem statement, your aim and objectives, and the literature survey. These three things are very much important for writing your synopsis, because that helps you to clearly account for a brief idea or a research problem and what you are testing. You might be testing the efficacy of COVID-19 vaccine. It might turn out good, it might not turn out good. So that is your testing hypothesis. You will justify. How will you justify? Through the literature survey, as well as the general connection that you have with the science and technology, with the inculcation of the habit of going through them. You can give certain methods and methodologies which fall into your testing hypothesis. Please don't give anything as such as the general ideology that you might do in a lab. Like say I took a virus, I gave it to an cell line, it produced an immunoglobulin, which I took, and it generated a very good amount of immunogenicity. No, you can't do that. You need to justify through methods and methodologies. Say, for example, considering the same thing, we would perform cell assay and analyze the apoptosis through certain techniques like flow cytometry, and all of these kind of specific methods that you mentioned will help you justify your hypothesis that yes, it might happen or it might not happen. Then you can also put in very brief assumption and restrictions of your studies, maybe the equipment or the your outsourcing reach capacity based on your geographical location, and all of that will help you to put it down in a short description, right? This is synopsis. Also, the one most thing that you should never forget to mention is the time schedule. The time schedule is very much important in synopsis. If you give to say that I would do this in 6 months, then definitely you should try and finish it in 6 months, right? If you don't finish, they will again ask you reasons. why you're not finishing, why you're not capable of finishing in that 6 months. If you give some silly reasons, you might lose the whole funding or half funding that you need to receive once you've been selected. So don't try and give a very much too expected numbers. Please do add an approximate numbers of the time schedule that you can do. And this is how you can present your synopsis in front of any research societies that is giving you opportunity. So if you have any questions, please do ask me. Thank you.
Thank you so much, Nitish. It was a really intriguing webinar for me also, and I was very happy to hear all the basic insights. I'm sure that the students here, the participants here are also are now very much that they also know how to go about it. And there are two questions that I can take right now. So Amrita is asking that since you mentioned that the abstract is better perceived as a summary, what's the difference between the abstract and a conclusion? Very nice question.
Yeah, the abstract is your summary of your whole study, right? The conclusion is when you consider your study, say, let's consider the antibiotic studies. In your antibiotic studies, you are giving a result that it is killing the organism, but your abstract is taking the introduction part of your literature survey that there is an expected possibility that the different species of fungus that you might have selected of Penicillium notatum might be producing the same antibiotic, but their molecular arrangement might be different. And when you treat them, the effectiveness is different. So that is the conclusion. So that conclusion will help you understand your abstract and make you difference. Hope you are understanding what I'm trying to say.
So basically, Amrita, so abstract is a shortened version of your entire proposal or a manuscript. So basically, for example, you are compiling your entire introduction in 1 or 2 sentences. Same goes with your material methods, and same goes with your results. And there's a small line of your conclusions. So conclusions is just conclusions, that what exactly are you concluding from a study. Abstract is a shortened version of your entire whatever you're writing.
Okay,
Hope
I
that
got it.
answers. Yeah,
Thank you.
most welcome. So Sanju has a question. He has actually 3 questions. So first of all, how to choose the field of research? One, let me take one by one. So how to choose the field of research?
So Sanju, the first thing I would suggest you is many times what happens is you need subject expertise or subject interest. Subject expertise is very much difficult at the bachelor's level. I do get it. But if you have interest, then the first thing that you need to do is proactiveness. For example, I know you might be intrigued of RDT part in your 12th and 11th grade, where they started to produce insulin using either a pig's gene or a cow's gene, and then came to the human gene, right? If that is intriguing you, then take the keywords, as I said, from that particular study, go onto the internet, start working upon those keywords. So they will help you to understand, is it really, the subject that you like, or do you want to change your research part? So that means you don't want to do RDT. If you don't want to do RDT, then you have always the open part where you can select the subject. But if this kind of statement from me is not helping to you, then the better option for you to select your field is just go to your peers, continuously start conversing with them. Okay, ask them questions: why you selected, what was the purpose for you to select, and how did you select, on what basis. So you will start getting answers like, I like the subject. The more intriguing part for me in the subject was the methods and methodology and their application part, which kind of suits to my ideology, that helps me work on this subject. So you will get something similar answers, which will help you to select your particular area where you would like to work on.
Okay, so the second question is a very sort of fancy question. So he's asking that, is there any possibility for artificial intelligence to automate life science research, or can it assist research?
So It's kind of very much intriguing, but it cannot assist. Human brains is difficult to replicate. It's not possible at all.
At least at this in this era.
Yeah,
Maybe
at least in this era, it's completely
maybe 30
impossible.
years down the line, yeah, we may be, but yeah.
Maybe
Yeah.
maybe with mathematical algorithms, it's possible. I'm not sure about it because I'm not ultimately good at mathematical algorithms. Maybe in future it is possible.
Yeah, but you can very well use artificial intelligence for your research. So that you can
Yeah.
use.
Yeah, for that, there are actually a lot of instruments currently in all the huge biotech companies where you don't have to do the single pipetting. The system does it for you. You just have to instruct it, keep the samples, keep your sample treating vials. It will do all for you. You don't have to sit there. It will do and it will leave. It's your job to put it into the incubator or other things. So that you will have to play your role again.
Exactly. So you can use our tools utilizing artificial intelligence like AI, ML tools for your research, but they doing your research is not yet possible. Maybe in future. Maybe you can work on it. And so the last question that he's asking is, what are the upsides and downsides of a research career?
Oh, that's actually a very good question if you ask me. The upside of research career is you have no limitation. You have no limitation. And please don't think that, many people do have this restriction. If I do a master's or a PhD in one particular subject, I am restricted to that subject the whole of my life. Please don't think like that. When I was in my master's university in Glasgow, I have seen researchers who have done molecular biology, but they got Nobel Prize just because they identified a new organism that could create a novel antibiotic. Where is molecular mechanism and where is antibiotic identification? So there is no relation with what their studies was. It's more related to new aspect and applications of your basic knowledge and information that you can apply. You can reach to any level. There is no restriction. The downside of the research career is that, when you do a biased research, or let's say nobody is giving you any sort of ideas, or you're not able to generate any sort of ideas, or you're just following the trend of bachelor's, master's, and PhD. After PhD, if you don't do a postdoc, then you will just end up doing a good job with a good salary. If not PhD, then only master's. Then it's very difficult because either you have to get experience. If you're master's, you will not get experience anywhere. It's very difficult to gain experience. I am the one of the very good experienced person in that place, because to approximately tell you, after my master's, even with the abroad masters, I have at least applied for 36,000 applications where I didn't get job. The reason when I get back to them was because my degree is very fancy and very much futuristic. It's not close to the current trend, what is happening. So it comes to what you're choosing in your subject as well. So please choose something as biotechnology or botany or zoology, or maybe biochemistry. Genetics as well is good, but don't go too much fancy like molecular biology, or maybe say, nanotechnology. Nanotechnology is good. I'm not saying it's bad at all. It has way range of good applications, but for that they will ask you experience, right? So all of these are downsides, and you might not end up as you expected.
Yeah. So Sanju, I also have same comments that the upsides are that you can get a lot of opportunities for your career. You have a lot of options at each stage. At a bachelor's level, also you'll get a lot of options. Especially this pandemic has has shown that what is actually the importance of research, and a common layman is also looking forward to us that yeah, we'll create miracles and we'll give them the vaccines. So that is the upside, that now initially only the doctors, I mean the MBBS or the practicing doctors were considered the bhagwan. Now everybody is looking up to the scientists, at what they are doing. The downside that I would like to tell you from my experience of being a PhD and a postdoc and a scientist and then an entrepreneur is that it takes a long time. So
Yeah.
if you would have been doing MBA or any other degree, you would have, and the money that you get is not that much. That's the only thing. But if you enjoy doing this, there's nothing else that you would like to do.
Yeah.
And I don't think there is a limitation for jobs also in this current stage. You can get a lot of opportunities. The only downside that I will say is that it will take you some, if you are planning for a proper research career, then you should do a PhD and something like that. So it will take some time, or you have to take experience. So I have seen friends like Nitish is one of the best examples, that he has his masters and he has got a lot of experience. And then I know people, I know friends who are either BTechs or masters, and they have got industry experience and they are doing good. So both pros and cons, and these pros and cons are in each and every field. You do anything, you go for engineering, you'll get this.
Yeah, but actually, one more thing I would like to mention is that Diksha is one of the best person for you to get to know about bioinformatics. Because as she was mentioning about the vaccine, the reason that they were capable of making it within 1 year is because of bioinformatics.
Exactly.
And nobody knows the importance of bioinformatics till recent pandemic situation. They were just ignoring bioinformatics to a very large extent. But now you can actually know that, bioinformatics helps you to speeden up all of the processes.
Yeah, otherwise they would have taken like 8 to 10 years to get the vaccine. And just imagine 8 to 10 years without the vaccine, what would have happened to the world? Forget about our country. Just-
Completely
yeah.
figured out.
So Surya is asking this question, that if possible, can I use my UG project as my research paper? I
Yeah,
think
definitely you can.
so. Is there any other question, or we should wind this up? We are already way past our scheduled time.
Yeah, timings.
If there are no more questions, I would like to take this opportunity to thank you, Nitish, once again for this wonderful webinar. And I would like to thank all the participants who have attended this webinar and asking such nice questions. And regarding your certificates and recordings, you'll be receiving both of them within 1 week. And if you have any questions, or if you would like to discuss on this more, or if you would like to have a small workshop on this particular subject or any other subject, feel free to write to us. We'll be very much happy to cater to your needs. And so thank you so much, Nitish. And Vismita, are you here? If I can take a screenshot, if you can switch on your video. Yeah.
Yes.
So this is a webinar. I won't be able to take the screenshot of our participants, but I am very much happy that we have got such nice crowd and such nice participants. Thank you so much, everyone. Have a nice evening. And see you all for the next.
Thank you, Diksha. Thank you, Vismita.
Thank you.